First, I would like to thank the organizing committee to give me the opportunity to present this work that is a collaborative work with 3 laboratories in France. For those that wonder where is Poitiers in France, it's this one. It's there at 100 kilometers from the west coast. It's a classical Roman middle-age town with Roman architecture, and also we have some a leisure park called the Futuroscope, which is involved in using latest technology to entertain people. And this is our brand new lab we are in since last summer. It's very nice. First, I will present the application we were working on, which is mesothelioma. There is several types, and the most important one is pleural mesothelioma, which is resulting from asbestos exposure. There is various risks of contracting this disease. In France and in other industrialized countries, this is mainly due to professional activities for men and to a smaller extent to women. The real problem is that it is a very poor prognostic disease with 1% survival at 5–10 years. There is a lot of clinical trials that are trying to find a solution. The first-line therapy is Alimta with cisplatin, but none of them are really effective. More recently, it was demonstrated by gene profiling that epigenetic aspect of this pathology could be investigated with some inhibitors. I will talk later on the various targets. The 2 main epigenetic targets used to study alternative clinical trial are HDAC inhibitors and DNA methyltransferase inhibitors, alone or in combination with other compounds. And you have here some representative examples of HDAC inhibitors, which are mostly hydroxamic acids, carboxylic acids, benzamide, and this is a cyclic peptide with a disulfide bridge. So epigenetics, for those that are not very aware of, are mechanisms regulating gene expression without DNA sequence modification. So you have here the basic unit of the chromatin, which is called the nucleosome. It's an octameric structure with DNA wraparound, and you have in this part you have histones, which H3 and 4 mainly, that have an N-terminal tail going out, which is a site for several chemical modifications, which are the basic principle of this regulation. And the 2 major epigenetic modifications on histones are methylation and acetylation, equilibrated by methyltransferase demethylases and recognized by some other type of proteins. For acetylation, you have histone acetyltransferase and histone deacetylase, and the acetylation level is recognized by bromodomains. So you have, in fact, 3 types of protein around there. You have the writers, which are putting something on histones or DNA. You have the eraser, we are removing this chemical group. And you have the readers that are able to read this. And this is what is called the histone code. And the combination of these several marks are the principle of the gene regulation, the regulation of gene expression. In the lab, we were interested a couple of years ago to design some HDAC inhibitor for zinc-dependent HDAC inhibitors. We were inspired from this natural product that was not very practical for development due to stability. And we went up after some work with this one, where you can see just a ring I did here with an oxygen, which is a nanomolar compound. And we are currently continuing to work on this. And in particular, the latest interesting result published in here was the reduction of resistance in mesothelioma compared to cisplatin you have here. And when you use a combination, you have cell variability, which is highly diminished. For some reasons, we were also interested in designing drug delivery system, the central focus of my talk. As you have seen during these 2 days, there is a lot of metabolic activities that is diminishing the activity of compounds by several ways. And in cancer, it's also a particular problem. You have a flux pump, you have a lot of mechanisms which make your compound not with a non-optimal activity. There is also a problem of selectivity regarding tissue cell targets. Oh, this one is in French, sorry. In the lab, the solution we are interested in are prodrugs or targeting, and I will speak mainly of targeting. Targeting, you have passive targeting or active targeting, and the third level of targeting could be the cellular compartment targeting, which is interesting in epigenetics because some of the targets are in the nucleus, some of the targets are in the mitochondria, and others are active around the tubulin and other elements in the cell. So how does this work? The passive targeting in cancer is mainly based on physiological differences between normal tissue. You have a normal vessel with a limited porosity, so you have small molecules that can go through. It's red dots in the scheme here. When you have a tumoral environment, the vessel, the endothelial cells in the blood vessels are less tightly joined, so there is higher porosity which can be used to deliver specifically macromolecules with some limited sizes anyway. And when you are in the tumoral environment, then you can go into the cells by various mechanisms. Some have been shown just previously, mainly endocytosis mechanisms. And we were interested in this work to use the acidification during endocytosis to release the compounds from our nanoparticles. So as I explained, it is a collaborative work with the Laboratory for Organic Polymer in Bordeaux, that have done all the polymer part. In Poitiers, we have designed some functionalities to attach on the polymers. And in Nantes, they develop everything around the biology of the objects. So our aim was to construct a generic macromonomer that could be functionalized by the popular azide-alkyne click chemistry. Our job was to do some functionalities with alkenes, and then we prepare a library of macromonomer, functional macromonomer, copolymerized, and the system used for the copolymerization generate core shell particles. So the chemistry beyond is this one. Here you have a peg chain, the yellow. You have here the function that can be modified to azide. You have here a norbornene group. This group is used for the polymerization process. Regarding selecting this type of macromonomer was important because you have polyethylene oxide, which are known to be to introduce stealth properties, which is important for the passive targeting to avoid macrophage capture and so on. And it is also important to stabilize the system during the polymerization. And the norbornene group is used for Ring-Opening Metathesis Polymerization with the groups 1 catalyst. Regarding function, we use this known rhodamine clickable dye, and we also develop this pH responsive system to be clickable, and I will come back on this later. Then we click this with that, we obtain this library, and then from this we can prepare various systems, mono or difunctional. At that time we have only difunctional and monofunctional. And the particle size here is spherical and homogeneous in size, and it is 300 nanometers. It is supposed to be a little bigger than what is normally used for passive targeting, but we will see that it is anyway working. So first we have demonstrated that our particles were able to highly selectively target tumor in vivo. We use this one with the reporter, and after 1 week you have accumulation of the particle in the mice, and this is the organ obtained after euthanasia, and you have here the biodistribution over time, and you see that there is high accumulation. You have some remaining fluorescence in the liver, which is always difficult to eliminate completely. You have in the blood a maximum after 6 hours, and then it vanishes, and you have some remaining results here also. Regarding the releasing system, as it was shown previously, we use the acidic pathway of endocytosis. So this is a simplified view of the system. If you have a molecule with a therapeutic agent loaded in, you can use internalization processes going to endosome at pH 6, depends on the literature. And then you have here maturation to lysosome, more acidic, pH 4 or 5. Here you can degrade the vector if it is designed for degradation under pH, or you can just release your compound by some pH responsive link on the particle, and then your molecule can go in the cytoplasm or in the nucleus, or where you want it to go. So we first, of course, verify that our system was able to enter cells by endocytosis and through acidic vesicle. This is classical stuff. You have here the fluorescence of the rhodamine part. You have here the lamp one, which is a marker for acidic vesicles. And if you merge, you have a nice colocalization. We have also demonstrated with the cytochalasin D and temperature that it is an active process, but we currently don't know really which one it is. Regarding the pH responsive system, we were inspired by previous work from Patel, and we focused on this trityl system, which is known to be a acidic pH cleavable system in protecting group chemistry. And our idea was to innovate in doing this trityl analog with another click chemistry reaction. And in fact, we end up with this kind of system, which in fact has 2 click sites to be used. The principle is that if you protonate at acidic pH the molecule which is covalently linked to the system, you will have a carbocation, which then can be trapped by water to give the corresponding alcohol, and you release your molecule. And then if you mix this with the monomer, the macromonomer, which is in the azide form, you will have this functional macromonomer to be used for copolymerization. Before that, we have validated the use of this system with these 3 HDAC inhibitors. These 2 ones are well known. This is CI994, this is vorinostat. They are all linked by this position. And this one is one we have developed in the lab, which is a nanomolar inhibitor inspired from the trichostatin I showed previously. We were able to scale up the synthesis of these prodrugs here, and we identify various We prepared various derivatives depending on R1 group, which is important to modulate the speed of the release, and which also depends on the type of functional group you use to link the system to the molecule. So there is 3 candidates here that were quite rapidly hydrolyzed at acidic pH and moderately stable at the physiological pH. This was the best we get, and we went on with that. For Vorenosta, we prepared this system with here OH only or the rhodamine reporter. We verify with cell system that there was some, I don't clearly see. We verified the cell viability. This is a nanoparticle with no functional group on, and this is the particle with the SAR. So there is an interesting result, not too high, but interesting. We verified that we have apoptosis in cellular assay. So the control, the particle without compound and the particle with SAR. So there was a nice response. And more than this, we also verified that introducing this system doesn't modify the biodistribution we showed previously, and this was the case. And we also do biodistribution studies on the cellular model. And then we try to see if there was some interesting result regarding a biological effect. So here you have the response regarding reacetylation of histone H3, which is shown with our nanoparticle with Vorenosta and not with the other control system. We have also checked for toxicity. There is no apparent toxicity. And then we use our nanomolar compound, which was linked to the prodrug system, which was linked to the macromonomer, which was then copolymerized. We have verified with a breath assay that there was effectively acetylation in cells. And then we tested this system. There was various group of mice. We tested various concentration of compounds alone or nanoparticles. And then this was made on an orthotopic model of mesothelioma that time in combination with decitabine, and we observe this nice biodistribution and tumor reduction with the particle 80% around. And here you have some images of pancreas that here at control, treated with a compound alone and treated here with the particle. You see there is no more cancer cells in this organ. So in conclusion, we have a clickable prodrug. That was applied to HDAC inhibitors, but we can apply to other molecules. We have a flexible synthesis for our DDS system. We are looking for industrial partners to try to develop. And we are now also going to inorganic nanoparticles with several projects. And I will thank all the collaborators on this project: biology, particles, molecular modeling, and of course chemistry. Thank you for your attention.